Okay, welcome everybody to session 2 of iNAV 2020. My name is Joshua Jacobs. It is my privilege to be helping involved in organizing this great conference, and I'm really excited about the session 2 on the topic of memory and navigation. Before we get started with the content for today's presentation, I have a couple of little notes that I wanted to pass along to everybody. Number one, at this meeting, we're doing a lot of challenging things in an unknown way during the pandemic using this crowdcast system. And I wanted to acknowledge that this meeting absolutely could not be happening without the help of the backstage crew here. There are 5 people sitting backstage who are essentially the directors for the event that you will see today, involving how crowdcast works, moving people online and offline and whatnot. Tamara Gedankian, Cameron Holman, those two people are graduate students in my lab who are very helpful with all this. Marzia Ferradoni, Jonathan Shine from the Jonathan Wilbur's lab, and Marie Vericel from Sylvia Worth's lab have all been extremely helpful with moving everybody back and forth, and I wanted to acknowledge their help in making this production go smoothly. So thank you to the 5 of you. We really appreciate your help with all this. The second note is, as a reminder for any Data Blitz presenters, if you have not emailed your slides in, please do so as soon as possible. We need to do a little bit of processing of all the files to make the Data Blitz go very smoothly tomorrow and Wednesday. So please email your a PDF version of your slides to mail@inavsymposium.com. Okay, let's turn to the fun stuff. There are 4 wonderful speakers that I have the privilege of introducing and moderating today. The first speaker is Andre Fenton from NYU. Following that will be Timothy Behrens from Oxford, Stefano Fusi from Columbia University, and Alcino Silva from the University of California at Los Angeles. Each presenter will be presenting for a total of 20 minutes, including Q&A, so probably a 15-minute or so presentation. And to all the presenters, if you didn't miss this notification earlier, I'll be letting you know when there are about 5 minutes left in your presentation, and I'll be handling the question and answer so you can focus on your slides and not have to worry about the details of the crowdcast system. I think that's about all the logistical information that I need to mention, and I look forward to a nice symposium. So we'll now pass things to Andre Fenton.
Thanks, Joshua. It's not often I get to say this, so I'm going for it. Good morning, good night, and good day, everyone. Thanks to the organizers for inviting me. I've actually never been able to attend an iNAV due to other duties, usually during the summer, directing a course in Woods Hole. So I'm especially happy to participate. You know, one bonus of the pandemic is that the sessions have shifted, and so there are a lot of us that can gather. So I'm always an optimist, looking on the bright side. So thank you, Joshua, Jeff, Thomas, Laure, Sylvia, and Mayank. I'm going to talk about, and I'll just switch slides now. I'm going to talk about work largely done by a senior postdoc in my lab in Hay Park, in collaboration with Jim Ronk, who helped us enormously conceptually in this. He discovered head direction cells, and I'll be focusing on head direction cells. Stephen Keeley, when he was a graduate student in the lab, very talented. He's now a computational postdoc at Princeton. And my colleague, Christina Savin, in the Center for Data Science. So we collaborated, and what I'm going to try and discuss today in this 15 minutes is really the question of, are the spatially tuned cells that that the modelers are using, and that many of us in this symposium study, are these cells in the hippocampal formation and the entorhinal cortex, are they components of a unitary representation of the space that's being navigated? And to use the phrasing in the Nobel Prize citation, are these really cells of an inner GPS of the brain? I don't have an answer for that, but I want to probe that with the experiments that we'll discuss. So in many ways, we've conceived of the navigation system and the way we've approached studying it, as you heard, in fact, Alessandro Treves challenged that a little bit, is very much like the GPS navigation system that we are familiar with. There's a constant a directional origin. There's always a north. Humans even invented a compass to make this absolutely clear and uncontroversial. But we only invented that approximately, say, 20 or so BC. And in fact, it was used, 200 or so years BC, and it was used until for about, say, 1,200 years to actually do fortune telling. And it wasn't until sometime in about 1040 or so that was started to use it for navigation. You'll see why that's relevant in a moment. This is Jim Ronk, and because we almost never record a single cell at a time, I'm going to play a movie from my graduate student days of a head direction cell. These are pretty remarkable. You'll see that when the head, I'll stop talking, right, the cell really fires when the animal's head is pointing in that particular direction here, in this case at about 11 o'clock, and it almost fires on no other times. One more discharge. And so talking about these cells allows me to have some clarity and certainty about their navigational tuning. In order to understand what I'm going to describe, and in fact, we couldn't understand the data for years as we looked at it until I remembered that Jim Ronk had given me this book, We the Navigators, about how the Pacific Islanders, in particular the Caroline Islanders, who are extraordinary navigators, but they do it without a compass. And the way they do it is complicated, but one of the things that they do for their directional navigation that we needed to recognize in order to understand the data I'm going to show you is they use this system called ETAC navigation. It works by not using a compass the way we would recognize you'd use a compass. They use the constellations. Where they happen to be, there are about 12 or so stars that more or less are constant in the horizon. And the navigator lines up some reference island. It can even be something that they imagine with one of the stars here, the star second from the right. And they start navigating until that star is no longer lined up with the reference island and the next star is. And then they change their directional system, and they do that for the next stage of the navigational journey, and the next stage, and the next stage. So in a sense, their compass is changing from each of these stages. It's only intermittently anchored to a particular set of features in the environment. And you'll see why that becomes important when we study what the cells we actually were able to record do. The cells we recorded, we're using them or recording them from the hippocampus, the entorhinal cortex, and subiculum now, during this two-frame place avoidance task. The area marked red and the area marked blue are marked red and blue for you and I to see them. The rat can't see the red and the blue marks, but it can recognize where it is and where the blue place is and the red place is using external cues. In particular, the red cues, or the red place is marked by the cues that he can use in the room, that are room anchored, that he can look around the room and see. And the cues that allow him to recognize where he is on the floor with respect to the rotating blue spot are the cues that he lays down on the arena surface. Smell, feces, urine, that kind of thing. And what's important about this, you'll notice that the rat is actually avoiding both of these places. Because if the computer detects that the rat has entered either the red or the blue area, it will electrify the floor mildly. The rat doesn't like that. You can see it navigates this very well. So we can train animals to do this task. They learn it rather quickly here. You can see in two conditions, with the arena stable and the arena rotating. It takes them about 4 sessions to master this. And we're going to look at the recordings after the animals have had 10 sessions and have absolutely mastered the task. Sometimes they won't even enter the either shot zone in about 30 minutes. So we're absolutely sure that they know how to navigate. And previous work has shown this requires the hippocampal system in order to accomplish the task. So we've recorded cells from the hippocampus, from the superficial layers of the medial entorhinal cortex, as well as the subiculum. But for this talk, I'm going to just focus here on head direction cells recorded in the superficial layers of the medial entorhinal cortex. So what do you see when you record those cells during this task? Remember, the animals know what they're doing here. If you look at the first column here, here are 1, 2, 3, 4, 5, 6, an ensemble of 6 head direction cells. They all look very sharply directionally tuned. We can record them also when the animal is on the rotating arena and doing the task. The waveforms are stable, but what you'll recognize is the directional tuning is lost, both in the room frame and in the arena frame, from both points of view. And yet the animal, remember, is navigating perfectly well. When we stop the rotation, the cells pick up their directional tuning somewhat, although sometimes the directional tuning has shifted, in particular where it's anchored or where it's oriented in the room. So this is remarkable because these cells, as we've heard, one would imagine are necessary for the animal to orient in this space and to do this complex navigation task. If you look at other electrical features of the hippocampus or entorhinal cortex, the local field potential, power spectra doesn't change, the firing rates are more or less constant, only the directional tuning is disturbed during this situation. And we can even decode the activity. We're recording Ensemble, so we can decode where the animal is using a Bayesian system, or in this case, a fit. And so you can see that here, when we look at the activity recorded during the stable sessions, you can compare it to where the animal was observed. We can decode the animal's direction very accurately using this system. However, when the arena is rotating, that becomes different. If we are trying to decode where the animal is in the rotating frame, the arena, we're very, very usually wrong. And you'll notice that the decoding here, where the animal is during the in the room frame, is actually not very accurate in detail, but more or less in general. And the animal's actually changing its direction gradually or quickly, whereas the decoder is missing all of those transients while the arena is rotating. So this was perplexing to us. We wanted the system to actually track where the animal was, but we were unable to do that. So we at some point decided to look at how are the cells actually firing. Let's just give up on trying to understand their spatial tuning. What does the activity of these cells look like? So what we did was we looked at pairs of cells and how they discharge together. If you look here at these 3 cells, you can compute a correlation amongst any pair of cells, and we did that for all the pairs. And we could plot on the x-axis here what that correlation was. Some cell pairs are positively correlated, some are negatively correlated on time scales from 40 milliseconds to a second. And on the y-axis, we made the same calculation, but when the animal, when the recording was done while the animal was doing the second navigation session with the arena stable here. And what I'll show you now is when the arena was rotating. And you'll see that how the cells fired, one with respect to the other, is exactly the same. It's indistinguishable whether the arena is rotating or the arena is stationary, although the spatial tuning is totally disturbed, as you saw. So puzzling over this, we tried to understand what could be going on. Here's just looking at what you just saw were all the head direction cells. Here's if you look at all the medial entorhinal cortical cells that we are able to record. Again, the system seems to be indifferent from our point of view here, looking at how the cells discharge, one with respect to the other, indifferent to whether or not the arena is rotating. It's very reliable, but the spatial tuning is disturbed. So we considered the possibility that maybe—
Andre, about 5 minutes left.
Thank you. That maybe within the animal's mind, if you will, within the network of activity, the animal was tracking direction. It just wasn't reliably anchored to what we thought it should be anchored to, which is the stable frame of the, or inertial frame of the room, or the rotating frame of the arena. And so what we did here was we estimated an internal direction. The details are in the paper here, but what you could basically say is, I'm going to pick one cell and make that cell the origin of the compass heading, if you will. Very much like the ETAC navigation. I'm going to pick one cell and use that as the current, if you will, orientation beacon, use that as the current star, and compute the directional activity, the directional tuning relative to that cell's activity. And what you can see here is if we do that, when the arena is stable, we get the stable activity, just sort of feasibility proof. And when the arena is rotating, now the directionality returns. In this case, you can see the directionality is preserved in the arena frame, not so much in the room frame. This is an actual unusual recording. That's why I show it. What we typically see is the arena frame is disturbed or distorted, and the room frame activity is maintained. But we can observe either case. So this shows that internally, the activity consistent with the temporal coordination that I showed you is self-consistent and maintained during the navigation on the rotating arena, letting us breathe a sigh of relief. So you can see us trying to quantify this. There's really no difference between the rotating and stable conditions. Another interesting fact is how long does the activity stay organized? So what we calculated here was the directional tuning of the cells individually and in the paper as an ensemble. And you can see that you could pick at a given moment when you chose the activity of a particular cell firing, how many seconds out in time would the activity of another cell tune to where the animal's moved its head since taking a fix, if you will, from the activity of the first cell. You can see that when the activity is recorded in the stable condition, the green dots here, that stability lasts a rather long time. It's self-consistent for out to several tens of seconds. However, if you look at the activity during the rotation, you get a different picture depending on which reference frame the animal is using. During the rotating, in the rotating reference frame, you can see that is also maintained, but the animal after about 10 seconds decides to, in some sense, ignore or no longer track its location on the rotating arena, at least in general. So it takes a fix, it maintains that fix for 10 or so seconds, and after a while that fix is lost or no longer maintained in the arena frame, in the rotating frame, but maintained in the larger inertial frame. So why is it that we couldn't see stable rotating, stable directional tuning during the activity? Our decoding shows us in this summary plot, the color maps here are where the animal spent its time. The dwell map, directional dwell map here is in black. And you can see that there's a minimum of where the animal, what the decoding error is, right around here and here. And those places correspond to this edge, if you will, of this curtain, as well as a landmark that may be hard to see in the image here, but over here. This corresponds to that location or that angular direction, and this corresponds to the edge of the curtain. So and this is in the room frame. What the animal seems to be doing is taking a fix, often at the edge of this this cue or this cue, and then for about 10 or so seconds, maintaining its orientation with respect to that as the cells discharge, and then it takes another fix, another 10 seconds or so later at either this location or this location most likely, but obviously other locations. So I'm just going to wrap this up. Let's get back to our question. You know, are these spatially tuned cells in the hippocampus, are they components of a unitary representation of the space that's being navigated? Well, if you look at the activity of these cells in time, as I've plotted here, and in this plot, the red corresponds to the system signaling the animal's direction very well in the room frame, and blue very well in the arena frame. You can see that activity switches back and forth. This ensemble prefers the room, So there's this one prefers the arena, but activity is switching back and forth. And importantly, this green trace here shows that how close the animal is to one of the shock zones is anti-related to where the directional system is anchored. And if you look at the bottom plot from an earlier paper, it's the same plot, but for the CA1 cells, actually activity is very strongly correlated with how close the animal is to one of those shock zones. So the entorhinal system is in some sense using the activity in the entorhinal system is anti-related to where the animal happens to be close to in terms of needing to avoid one shock zone or another, and in the hippocampus, the opposite. So how might these cells be coordinated? And I'll just finish up here. If we go and we look at the tuning of these cells, how they respond one cell to another internally, ignoring the directional tuning or any other spatially tuned variables, you see in the entorhinal cortex and in the subicula, the activity is extremely reliable in terms of its temporal coordination, not so much in the hippocampus. And if you look at the hippocampus and its relationship to cells in the medial entorhinal cortex, whether they're head direction cells and place cells here, or just all pairs of cells that we recorded in the two areas, they're very poorly, reliably coordinated, suggesting, now I'll get to the take-homes, suggesting that if you look at the activity in these cells in these areas in isolation, they seem to represent the environment, but the directional tuning is clearly dynamic, at least in the entorhinal cortex. This neural coordination and the representational dynamic switching from the room frame to the arena frame seems to be distinct in different parts of the system. And that really begs the question, how is it that these neurons, these spatially tuned cells in different areas actually can be coordinated in order to navigate, to build a single navigation system? So thanks for your attention. Here's my funding, and I'm happy to take questions.
Andre, thank you very much for a very nice talk. There has been a very substantial conversation in a chat field asking about ETAC navigation, and I think the most useful thing you could do with the question and answer session would be to explain ETAC navigation and to explain how that may or may not relate to head direction cell patterns you've been seeing. That's kind of my reframing of a question by Jean Lorenz.
Sure. So ETAC navigation is a navigation system that's ancient, that has been used by humans for a long time, and it's used because there was no compass for a long time of human history. The way it works is cartooned here, and if you go to this YouTube link, I made a little video that describes it. The idea is that you have a variable compass sense, if you will. For some time, you take a fix between some landmark in the environment, and the expert navigators are capable of imagining this place. And so you line up your own position with this reference that you can physically see or imagine, and some celestial body that's going to serve as the anchor for the directional system, a star. And then you can start moving. You can start paddling your canoe. And as you paddle your canoe, you'll move, and eventually you'll notice that the reference thing that you've imagined or you're looking at no longer lines up with that star, it lines up with another star. So you can imagine this is like taking a fix, then doing path integration until you take another fix. However, the fixes are not determined by these salient, constant features of the environment from the point of view of an external observer, like an experimenter. They're determined by the salient stable features subjectively by the navigator. And that's what we believe we were able to see when we looked at the place cell activity and the head direction cell activity, as I've shown you. The system seems to take a fix, and for about 10 or so seconds, navigates. Relative to that, then it takes another fix, perhaps not on a star, but on another feature of the environment. Then it can take another step and take yet another fix. In the ETAC navigation system, the celestial bodies are exchanged systematically, not necessarily so in the rodent navigation system. And this is a navigation system that we can use to explain the data that we observe. I hope that answered the question.
That was great, Andre. One more quick question before we move on to the next talk. There's a question by Christoph Koch, who asked, could the rotating area on the ground possibly be used as a cue guiding head direction cell alignment?
So we actually believe that it is, and if you do the right kinds of experiments, for example, you could put water on the arena surface so that the animal can't orient to the cues that we know are important to it, which are olfactory. We would expect to not see this kind of reorientation to arena cues, but we would expect to see exactly what I showed, which was orientation to the distal cues in the room. However, putting water on the arena surface and turning off the lights would force the animal to have to use what was available to it, which would be its own internal sense, and it wouldn't have any stable visual cues to anchor to, and nor would it have local cues to anchor to. And when we do that, we notice that animals can only navigate moving approximately 7 or so meters before they appear to be lost.
Great. Okay. Thank you very much, Andre. I think that's it. So thank you very much. We're going to move on to our next speaker now, Dr. Timothy Behrens.
Hi. Hi. Oh, sorry. I might just turn that light off and put a different one on because it's right in everyone's faces.
We can't actually see your camera right now.
You can. You can't.
We cannot. No.
Is that on?
Not yet. Sometimes it takes a moment, but not yet.
I can see my face on. Can you see my face?
No. Other people can.
Okay. Well, let's see if not.
Apparently, other people are saying that they are able to. So I'm the outlier. So you can.
Okay. Good. Okay.
Go ahead. Why don't you try and share your screen? I know what you look like, Tim. So that's all right.
Okay. Cool. Okay. Good. Can you see the? Can you see?
Can anybody?
If I go. If I do that, can you see my slides?
Can someone tell me if they can see his screen? I think it's coming, and I see your face now too. There's some delay that was longer for me.
Okay, I can see my.
No slides yet. Screen share window is black.
Whoa.
Maybe you can do a whiteboard version of your presentation.
I could try. I'm just trying to. I can try. This worked on the.
There we go.
On the test. Ah, okay. So I need to do the whole screen then. So sorry, guys. This worked on the test on the test earlier, but
no problem. No problem.
It didn't work here. Okay. So now can you see the actual whole screen now?
Yes.
Okay. So I'm going to have to play this in the whole screen. Hi. Thanks everybody. Sorry for that. My name's
Take
Tim.
it away.
Hello.
Go ahead.
Okay. Cool. My name's Tim. I work at Oxford in London. Thanks very much for inviting me. I've never been to iNAV before either, and it's a fab conference. So I'm going to talk about how many spatial hippocampal things can you predict if you assume the hippocampus only does relational memory. And this is work by James Whittington, but it's really related to work that Stefano Fusi is going to take on a similar theme in the next talk. Okay, so here is a simple model of memory. Everyone knows it. It's just Hebbian memory. So it works like this. You stick a sensory representation onto a neural network, and the weights between the activated neurons, the synapses, strengthen. And this means that later you can provide a portion of that input, index the network with a portion of that input, and the activity will flow through an attractor in the strengthened synapses, and will retrieve the rest of that, pattern complete, retrieve the rest of that memory. Good. Oh, sorry. That wasn't what I was trying to do. Right there. Okay, sorry. Is that better? Okay, good. So this is a very simple model of memory, but it's been used a lot to model hippocampus. There's one way in which it's simple. You can see here in this schematic by Mans and Eichenbaum. And in the Eichenbaum world, the memories are a bit different. So it's true that they do get a sensory input, but it's also true that they get this other kind of mysterious input from the medial entorhinal cortex, coding something like context in the Eichenbaum frame. And then the hippocampus's job is simply to bind the object or the thing in its context to make what they call a relational memory. So relational memory is a thing in context memory caused by binding different inputs to the hippocampus. Okay, so let's take this at face value and just change our attractor network to do this. So this is a kind of model I'm going to be working with. It's going to get a mystery input from the medial entorhinal cortex, a sensory input from lateral entorhinal, and it's going to bind them together just exactly in the same way as before, except now the cells only fire if they get both inputs rather than getting one input. So it's a conjunctive memory. Okay, so this is now a relational memory, a thing in context memory, but what is that weird thing in the MEC, that blue thing up there? Well, to understand what this is, it's worth thinking a little bit harder about what a relational memory is. And so here's an example. If I tell you that Janice has a brother called Bob and a child called Alice, then you, because you're clever humans, can tell me all sorts of other interesting things about this family. Here are some of them. For example, you can tell me that Bob is Alice's uncle. So you can tell me that because relationships have structure, have meaning, and this same relational structure is true for lots of families. And so if you learn a relational structure in one situation, then it's useful to you to be able to immediately generalize it to some new data, some new information, some new family in this case, when it comes along, because it lets you make these inferences of things you haven't seen, relationships you haven't seen. But to do this, you need a representation of this abstract relational structure that's separate from the relationship of from the representation of any individual family. And so that's what you need in a relational memory. And that's the kind of thing that this medial entorhinal code is going to have to provide if you're going to do good relational memory. It needs to know, where am I in this abstract relational structure? I'm over here in this family tree, for example. Okay. All right, so what's all this got to do with space? Well, let's now think about this simple prediction problem. If I tell you a sensory stream and the relationships between that stream, so light bulb, arrow, right arrow, brush, down, motorbike, right, etc., and then say, what comes next? You've got no chance of doing that unless these different arrows here have a consistent relational structure, unless they mean something. Okay, but you can do it if they mean something, as in space, because you've experienced many of these spatial worlds, and you've abstracted this structural regularity of two-dimensional space away from all of these worlds, and now you can just use these arrows and they're meaning it with respect to this 2D space to say, okay, the light bulb is going to come next. Okay, so that's in that way, spatial reasoning is really exactly the same as relational memory. Okay, so we built a machine that basically solves these kinds of problems, called it the Tolman Eichenbaum machine, for reasons that will be obvious to everybody. And this is what it does. It does exactly what you saw in those last slides. It tries to predict the next thing, and we give it lots and lots of worlds that have the same relational structure, the same kind of family tree structure or spatial structure. And by the time it's trained, it'll take one of these, it'll go into a new world and take one of these tortuous routes through a new world. And when it's seen all the elements, it knows everything about the world. It can guess all of these relationships that it's never seen before. It knows where it is inside the relational structure. So that's what it can do. Its job is to effectively guess links that it's never been seen, that it's never been shown. And to do that, it's going to we're going to train a bunch of weights. The only really important ones to understand the relational structure are these weights up here. But later on today, Stefano Fusi is going to tell you some other really important things about the sensory representations, which come from training these weights down here, I think. Cool. Okay, so I'm just going to show you, spend the rest of the talk mostly showing you what it looks like when you've trained these weights. Okay, so first I'll just show you that it works. And so this is it trying to learn a tree structure. And this blue line down here is how often it could guess the right link that it had never seen before, when it had never, for the first family tree it ever saw, and it was awful at it. But then when it's seen lots of different family trees, it's great, and it can guess every single link that it hasn't seen before. So you can show a bunch of different nodes. You could say, okay, Janice is Rob's aunt, and then and it can start guessing these other links in the tree that it hasn't seen before. Okay, so to do that, what are the constraints? What are the impositions on this representation? Well, there's two real pressures. The first is it should make different representations at each location, so that you have distinct memories at each location. So it's learning to use that attractor network well. I want to make things that are easy to remember. And the second thing it should do is whenever you come back to the same place, it should make the same representation so you can retrieve the right memory. This is kind of like a bit of path integration. But it's never going to learn path integration with like a head direction circuit, because that would be a really hard thing to tie memories to. So these two pressures will both fight against each other. Okay, good. So let's see what those pressures do to representations, and we're going to start by letting it just diffuse randomly around a two-dimensional space. And if you let it diffuse randomly, subject to those kinds of rules, predict the next thing that comes, then it likes to learn these kinds of representations. These are what it thinks are the optimal representations for indexing memories and predicting memories in two-dimensional space. And so it has grid cells. It has grid cells of different frequencies, and within frequencies it has grid cells of different phases, and it has band cells. So these are things that we often find in the entorhinal cortex. I don't have to tell you guys that. In the hippocampus, it learns place cells, and we know why it learns place cells. It learns them because it's binding together sensory events effectively with grid cells. And so it's going to go pick out some of the nodes that happen to be aligned, some of the peaks of grid cells that happen to be aligned to the sensory input that that particular hippocampal cells fancies. So you have grid cells and place cells, grid cells and entorhinal, and place cells and hippocampal, just from the requirement to predict the next memory. Okay, so what now if we let it behave more like a rat? Rats don't often move around by diffusion. So let's do something else. Let's say it likes walking around walls, or it likes approaching shiny objects. This changes the transition probabilities of the animal, so it should change the best representations that you would have to predict the next memory. So if you do that, then you start seeing cells a little bit like this. Here are two cells. One always fires 4 pixels to the north of this object, and it works for any object. So here there are 4 objects in this environment, and this one here always fires 4 pixels to the east of these objects. And these are a little bit like cells that have been called object vector cells that have been recorded in the Moser lab, again in the medial entorhinal cortex. Because it likes running around walls, it starts building border cells and border vector cells, again cells that are often found in the medial entorhinal cortex. So this is looking like this zoo of different apparently bespoke cells in medial entorhinal cortex is really just a basis set for representing the possible transitions and inferring them immediately in new worlds.
Tim, about a few minutes left, please.
Cool. So So now let's move into the hippocampus, and we can see things that look like those object vector cells, but they're a little bit different. So you can see this guy only fires for a couple of the objects, and that's because the hippocampal cells are conjunctions again between the object vector cells and the sensory input. And again, that's rather similar to what these landmark cells, the object vector equivalents, look like in hippocampus. So just to compare that to the object vector cells in entorhinal, which generalize across all objects in both TEM and in real entorhinal cortices. So this is something that I'm excited about because I'm interested in things like reinforcement learning. Because you've got this general relational structure, not just space, you can represent more complex tasks. Here's a fun one that's been published recently by Chen Sun, where a mouse had to run around a maze 4 times to get a piece of cheese. Well, the Tolman Eichenbaum machine says, well, you need a certain cells to do this task. You need some cells that always fire like place cells or like spatial cells, because you need to predict the immediate sensory input. But you also need some other cells that separate out which lap you're on, because you need to understand, you need to make effectively a larger map that takes you all the way to the reward. And in fact, there's a third. So here are these cells again. The laps are now lined up one after the other. Here are spatial cells, lap-specific cells, and it says you need a third class of cell called these counting cells, which gradually increase towards the reward. And if you go and look at the types of cells that are recorded, you see exactly these types of cells: spatial cells, lap-specific cells, and cells that gradually increase. But TEM says, because the Tolman Eichenbaum machine knows why these have happened, they say that in principle they're exactly the same as a whole bunch of other classes of cells that have been recorded in hippocampus. And so here's one fun one that we all know about. These are called splitter cells. When animals are doing an alternation task on a T-maze, they'll develop some cells that like going left and some cells that are like going right. And that's because, according to that's exactly the same reason as these lap cells that were in that previous thing, because you need to know where you are in this larger loop of going left, back, right, back, in order to predict when you're going to get a reward. Similarly, inbound cells, outbound cells, all of these things fall into that same latent state category. Okay, if I've got 2 minutes left, I'm just going to show something interesting about remapping. So there's an interesting puzzle by this kind of theory of what hippocampus is doing, and that is what we're saying is some structural relational knowledge is generalizing across many different environments to allow you to make really efficient inferences in your memories. That seems like an impossibility given what we know about hippocampal remapping, because what we think is that these place cells, where these place cells are in one environment, will tell you nothing about where these place cells are in another environment. So how can these memories retain the same relational structure? Well, first let's go and see if that happens in TEM, and it does. So here's world 1. So these are real cells over here, world 1 and world 2. It looks like they're just going to random places. And in TEM, same thing. It looks like it's going from here down to here in world 2, and this one's moving from here up to here. But in TEM, we know why this happens. It happens because although because in these different worlds, although it is getting the same relational information from the medial entorhinal cortex, or the same correlation structure from there, it's getting vastly different sensory information in each of these worlds. And these conjunctions are what makes the place cells. So the sensory stuff is different between worlds, but the structural stuff is preserved. Well, if that's true, then we can make a prediction, which is that when a place cell moves to another world, the place it's going to move to is going to be at the peak of the same grid cell, a different peak of the same grid cell, if you see what I mean. So if this place cell started at one of the peaks, it's always going to have to end up at one of the peaks. If it started at this trough, one of these troughs, it's always going to have to end up at one of these troughs. That will retain the relational structure across worlds. And so that means that I can plot something like this. I can look at the peaks in these two worlds, and I can look at the grid cell firing at one place field peak, and the grid cell firing at another place field peak, and it should correlate. And to cut a long story short, it does. It correlates in 2 independent experiments, and it correlates about as much as we would think it should, given the model. So that's me done. I think there's strong similarities between relational reasoning and spatial reasoning, and hippocampal remapping isn't random. That's what I think. Thanks. Oh, I should probably also just put these faces up at least.
Thank you very much, Tim.
Should I
get
rid of screen firing?
I can tell you, yeah, I can tell you the 2 biggest questions that we have time for. The first question from the audience is from Habiba Azab. And Habiba asked, studies have often found non-stereotypical place/grid cells that nonetheless have significant spatial selectivity. What role, if any, would those have in your model?
So I didn't quite hear that. So are you asking, is Habiba asking about things in entorhinal cortex that are not grid cells?
I think she's asking about what she wrote as non-stereotypical place/grid cells. I think she means cells without beautiful spatial tuning. Oh, yeah.
So so I think that there's an interesting question about how you should go about representing structure, and if things really factorize beautifully, as in you should have something, all of the worlds that you go into will have something, will respect something about 2-dimensional space, and so it's a really good thing to factorize that part of your representation of structure away from everything else. But there's loads of stuff. Once you think the grid cells are really representing transitions and tasks, then there'll be loads of fun things that should be represented there as well, separately. Like, for example, if animals have a little home place that they like in the boundaries, that will be coded in the transition probabilities. If there are some animals that only run one way around a track, or like running one way around one way around one wall, another way around another wall. Those kinds of things dramatically affect what representations you should see. And so I think that it's really within the bounds of this general idea of the medial entorhinal cortex having a basis representation of possible worlds, that it should have lots of cells that are definitely not grid-like. Yeah, exactly.
Cool, thank you. One last question from Mark Brandon. In this framework, why do you need head direction cells?
So I beg your pardon?
Mark Brandon asked, in your framework in TEM, why do you need head direction cells?
Well, so it turns out that the head direction cells are extremely important, because I'm looking for to learn the relationship between actions, but you have to know what direction you're going in. My thing is not providing the so the relations that go into, you saw those arrows, that's an input to my system, right? I'm not and so definitely we need those, and actions or aunts and uncles in the other relational problems. There's an interesting question about whether we might be able to do it without them, which I think probably we can, but definitely you can do a much better job of predicting memories if you know where if you can path integrate head direction cells to predict the next memory. You can do a much better job of it. I think that what a more interesting question is, well, are those entorhinal head direction cells really needed for navigation, or are they just needed for making efficient memories? Whereas maybe some of those beautiful thalamic head direction cells would be fine for path integration, and then really they're only in the entorhinal cortex at all because they're useful for predicting memories.
Makes sense. Yeah. Okay, Tim, thank you very much. That was wonderful.
Thanks, everyone. Sorry it was a bit stumbly and I had some tech problems.
It was great, Tim. Thank you. Have a good night, Tim. All right. Our next speaker is Stefano Fusi from Columbia University. Stefano, would you like to share your screen?
Sure. Okay.
Stefano's talk has a intriguing title: Are place cells just memory cells? Memory compression leads to spatial tuning and history dependence. So this is a different
Okay.
take on things.
Can you see my slides?
Yes, by Marcus Bennett and Stefano Fusi. Are place cells just memory cells?
All right. Thanks a lot, Josh. And thanks to all the other organizers. I really feel alive again when I attend these workshops. And I'm glad that I'm speaking after Tim Behrens, because his beautiful work is very much aligned with what we think about the hippocampus and the system in general. And you already prepared the audience for this important relation between spatial representations and memories. Also, I will focus on a slightly different aspect, and as you will see. So, our main idea is that the hippocampus is basically a memory device, and it's there to compress the information before it's actually stored more efficiently, maybe in the cortex and also partially in the hippocampus. And this is an old idea, but we formalized this idea, and we looked at the consequences when then you consider this system during a navigation task. Okay, so most of what I'm going to present today is done with Marcus Benna. We have a bioarchive paper if you're interested in the details. And we are now working with James Priestley and Attila Lozonsky to test all these ideas in experiments. And Lorenzo Posani and Jack Lindsay are extending the theory that I'm going to present today. Okay, so let me start with the memory component. This is just a simple representation of the Hopfield model, which was a very influential model. It attracted a lot of physicists to the field. And one of the assumptions he made, was that the memories should be random and uncorrelated. You need to make some assumption about the nature of the memories if you want to say anything about memory capacity. So this is very convenient because you can do calculations and it's relatively simple. However, we all know that in the real world, memories are not random and uncorrelated. They actually have a lot of structure. For instance, the kind of structure that Tim Behrens showed you earlier in his presentation. So, this seems to be a very gross simplification, but if you think about it, it's not totally unreasonable, because when you go through some sensory experience, what you want to do to store a memory is first to combine it with what you already know, so what is already stored in your brain, and generate a compressed representation. And if you do a really good job, then essentially the representations you end up with are kind of uncorrelated. Otherwise, it means that you didn't compress them enough because they still have correlations. And they're very similar to the random and uncorrelated memories that have been studied. However, somebody in the brain, some part of the brain should take care of this process. And, this is essentially what we studied. Okay, so this is a very simple compression process. It's MPEG. Many of you are familiar with it. Even if you're not, this is the main reason why we can have this conference on the web, because the videos are compressed. And if you look at multiple frames, you see that there are a lot of correlations between all these different frames. The background is almost always the same. So MPEG, what basically does, it's much more complicated than that, but just to give you an idea, it looks at the differences between the first frame, the key frame, and the following ones. And these differences have two interesting properties. The first one is that they're approximately decorrelated, and the second one is that they're very sparse. And so, if you can store only this, then you can actually store these memories of the different frames in a much more efficient way. Okay, so how do we formalize this problem, and how do we build a neural network that then is able to do this? Okay, so let's say we start from these very simple correlated memories that were introduced in the 80s, in the 90s. You start from a pattern, let's say a random pattern. I'm sorting here all the neurons. Each square represents a neuron, and I'm putting first all the active neurons and then all the inactive neurons. This is an ancestor in a tree, and then you can build the descendants by flipping the state of activation of some of the neurons randomly. Okay, so for instance, here I change the activation of this neuron and this other neuron. And then I can generate as many descendants as I want. They will all be correlated to each other and to their ancestor. Okay. Okay, so now what you can do is to live in a world where you have this very simple structure. You have a bunch of ancestors. They are random and uncorrelated, if you consider them. But then each of them has a certain number of descendants that are correlated to the ancestor. Okay, so P ancestors and K descendants per ancestor. Then what you can do to compress this information is something similar to what I showed you earlier with the MPEG. So what you could build is a representation where you're using a relatively small number of neurons to encode the ancestors, because the ancestors are much less than the descendants. And then you can encode the differences between the descendants and the ancestors. And if the descendants are similar enough to the ancestors, these differences are going to be very sparse. So that would give you a big advantage when you read out this information with a neural network, because there are many beautiful old works that show that when you sparsify the representations, the number of memories that you can store scales linearly with the number of neurons, and like 1/f, where f is the coding level or the fraction of active neurons. Alessandro Treves already related this to what happens in the hippocampus in the 90s. Okay, so now, this was just to give you an idea, but we want to do it for any kind of inputs, also more complex structures that you might have in the input, not only this ultrametric tree. So the model that we are proposing is basically an autoencoder, and you have an input here. You have a larger number of neurons in these intermediate representations, and then here you have the reconstructed input. So basically, the reconstructed input is there only to determine the weights between the input and the compressed representation. So it's a criterion for us to know, what are the weights that you need in order to compress efficiently the information so that you will be able to retrieve it at a later time. Okay, so let's assume that in the input I have n neurons, and in the compressed representation you have 2 n neurons. Now, given that you have more neurons in the compressed representation, you can imagine that, at some point we're going to map this to the dentate gyrus. So, you might have a lot of neurons there. So how do you compress? How do you encourage the system to compress the information? So one simple way, and this is also an old idea by Olshausen and Field, is to impose sparseness to the representations. Okay, so basically we have a sparse autoencoder that is going to be used to build these compressed representations. Okay, so first of all, let me show you that this really works, and it works extremely well. We were surprised to see how well it works. So let's say you build this system here, and now you want to count how many memory resources you need here. So it goes like 4n squared, where n is the number of neurons that you have in the input. You have 2n in the compressed representation. We're going to compare it to the case in which you read directly the input, and then you try to reconstruct it in this output layer. So here you have significantly less neurons and synapses. So, there must be a really good reason to go for the first model instead of the second one, given that the first one requires a lot of resources. And the third one will be like the first one, but with no training, where we just use random projections. Okay, so what we did was to use these ultrametric memories, and we progressively increased the size of the network here. So basically we change n, and we see what is the maximum number of memories that can be retrieved for all these 3 different models. And you see that the autoencoder, despite the fact that it's using more resources, in the end, when you compare the models for the same number of total synapses, it actually does significantly better than when you read out directly. Okay, so again, this is not trivial, because there is a lot of machinery here you have to invest on in order to build this compressed representation. The random case does significantly worse. This depends on the amount of noise you have in the input, but in all the cases that we analyzed, it's usually the worst. Okay, so this is a good memory model. So what does this have to do with navigation? So the idea is that the hippocampus might be this device that combines the sensory experiences with what we already know. It could be recent experiences, for instance, when you explore a completely novel environment, or it could be some knowledge that you already stored in the cortex, as Tim Behrens presented. And you want to combine these two different sources of information to build an efficient compressed representation. Okay, so now let's imagine that you have this little mouse that is exploring an arena, a square arena, and then it's going around. And what happens is that all these sensory experiences are going to be correlated to each other. They're not going to be identical, but they're going to be correlated. And in particular, when the animal goes through the same location, these two sensory experiences will share something, and they will be similar to some respect. So how do we take advantage of these correlations to store memories about this particular environment? Okay, so here we built this autoencoder, and we trained it. And we made a very minimal assumption that essentially in the input, we have information about the head direction, we have the information about the distance from the last wall that was visited, and we have the position of the last wall. It doesn't really matter exactly what information you put into the input. The only important thing is that you have some low-dimensional structure that can be used then by the autoencoder to extract this compressed representation. So there are a lot of other structures that you can have, which would lead to the same kind of response properties that you have in this case. Okay, so let's say that the animal is exploring. Now this is a simulation. The animal is exploring this arena with these strict trajectories. We simplified a bit. And then what we did was to train this autoencoder. And then if you look at the response properties of the neurons in the autoencoder, you see that they resemble very closely the kind of responses that you observe in the hippocampus, and in particular in the dentate gyrus. So, you see a lot of place cells. Some of them are nicely localized. Some of them have multiple peaks. You have these cells that tend to be active when the animal is close to the border. And the other most important property of these cells is that they are rather unstable. So, for instance, in this very simple example, you see that the animal, the simulated animal, is exploring this part of the arena more than this other part. Okay, so if these are really memory cells and they're just reflecting all this sensory experience, something about the statistics of all these sensory experiences, then they will be affected by any bias in the exploration or anything that happens in the world, so any salient event, the delivery of a reward or anything else. So for instance, okay, thanks. So for instance, if you now consider the same environment and you bias the exploration in a different way, so for instance, now the animal is exploring the upper left corner more than the bottom right, and now you continue to update the representations in your memory system, then what you see is that you get these place cells that are similar to the original ones that you got after essentially the exploration statistics A, but they're also different, and they're actually different enough that you would be able to decode whether you're coming from exploration statistics A or exploration statistics B. So basically, I don't have time to show all the details, but you can find them in the paper that I mentioned. What we show there is two things. So the first one is that it's actually possible to decode the position of the animal with relatively high precision, despite the high variability. Because here there is a lot of variability. Whatever happens in the environment or whatever happens in the exploration statistic of the animal, the response properties of the neurons are going to be affected. Despite all this variability, which is actually observed in a lot of experiments, and Andrea Fenton showed a lot of beautiful examples of highly variable fields, you can still decode the position of the animal. And the system is only designed to store memories efficiently, but when you are in this particular situation, it can be used also to encode position. So the second consideration I want to make, and it's also a prediction that we are testing in experiments, is that imagine that you have a sequence of exploration biases, something like AB, AA, BA, BA, and so on. Then by looking at the firing fields fluctuations, you can actually tell what it was the exploration statistics in the recent history. So, these are really memory cells in the sense that they're continuously updated and they reflect the recent history. And we can show in the paper that it is possible to decode the recent history. And again, that's what we're testing now in real experiments. Okay, so let me conclude. So correlated memories can be stored more efficiently by compressing the representations. The dentate gyrus is assumed to decorrelate these representations, as we did in this sparse autoencoder. And then, pattern separation is a natural consequence of this process of compression. Compression leads to spatial tuning. It's something that you get very naturally. And this spatial tuning has these two interesting properties. So the first one is this history dependence. And, I'm not claiming that this history dependence accounts for all the variability that is observed. There are clearly many other sources of variability, but we believe it could be an important component, at least in some situations. Nevertheless, these representations, at least at the population level, they are stable enough to allow for decoding the position of the animal. So the other thing that is a simple consequence of our assumption is that hippocampus would compress any type of memory, whether it's spatial or non-spatial. So for instance, there are recent experiments that show that even some kind of navigation in an auditory space would lead to place cells. So essentially, our prediction is that every time you have some low-dimensional latent structure in the input, that's going to give you a representation in the hippocampus that might be similar to what we observe when the animal explores some environment. And finally, remapping, for us, it's a simple consequence of the fact that, again, these are memory cells rather than place cells, and they're continuously updated. So they're affected by any new memory that is stored, especially the salient ones, like delivery of reward. Okay, the only thing that is really missing here is then, how do we incorporate this into the loop with the cortex? And, Tim Behrens showed you a beautiful way of doing it, and we are moving in that direction to incorporate our model in something similar to the system that Tim Behrens presented. Okay, thanks a lot for your attention.
Thank you very much, Stefano. I'm going to take advantage of my privilege as a moderator and ask you a question myself, if I may. How does this relate to neurogenesis, which is what I think of, and many people think of when they talk about the dentate gyrus?
Yeah, so it's something we're thinking about with René Hen. And, one of the ideas is that in our simple model, sparseness plays a very important role. And depending on the compressibility of the environment, so for instance, whether you are in a rich environment or not, you have a different optimal level of sparseness. So one possibility is that neurogenesis can affect very rapidly this level of sparseness in an indirect way through the inhibitory network in the dentate gyrus, and can adjust the level of sparseness depending on the richness of the environment.
Oh.
So that's one hypothesis we are now testing in some simulations, and Jack Lindsay is actually running simulations now, but it's too preliminary to really draw conclusions.
Cool. There was a question from Matteo. Could you clarify the role of ultrametric distances? For example, is there evidence that the things we or a rat may want to remember are arranged according to an ultrametric distance measure? Excuse me.
Yeah, so I mean, you shouldn't take that example too seriously. It's just one example that we can analyze, and we can do some analytical calculations, but it's really the simplest form of correlations we could introduce. So we never made the assumption that that's the statistics of the real world, although in some situations it might be. So, the autoencoder works with any statistics of the input. And in particular, the kind of statistics we used when we simulated the mouse exploring the environment, that's not ultrametric.
Okay. Thank you very much. We have a bunch more questions, but we're running a little late on time, so I'm going to move on to our final speaker. But thank you very much, Stefano.
Thanks, Josh.
Okay. All right. Our fourth speaker is Alcino Silva. Hello, Alcino.
Hello. Wonderful to be here.
Wonderful to see you.
I hope you can see my screen and my presentation.
It looks wonderful, seamless, great.
Wonderful.
I'm going to get out of here and let you give your talk. Thank you very much.
All right. Okay, this is going to be a change of pace. This has been an amazing session. And what I will tell you today is about mechanisms that link memories across time. And all of the new work will be work that Yang Cheng and Mio Zhou has done. And this is based on studies that Denise Kai and Daniel Aroni did in the lab initially. I was lucky enough recently to be in this amazing place in Norway, and I stopped and took this picture. About 5 hours later, I was in another amazing place just miles away from the first one. I hiked around this place, and it was astonishing, and I took this picture. Now, on the same trip, this is one of the lucky things about being a scientist. On the same trip, but about 7 days later, I lectured elsewhere, and I took this picture. And again, another beautiful, stunning place. But the question is, how is it that I know without any doubt that the first two pictures on the left were taken on the same trip in Norway and not the other picture on the right of the screen? How is it that we can link memories across time and separate them from others? It's clear that this is highly adaptive, but the question is, how do we do it? Now, a few years ago, I proposed a very simple-minded model that said that essentially, if you look at a region of the brain that captures memories such as this one, like, for example, the hippocampus, we proposed that a set of cells in your CA1 region, retrosplenial, in a lot of the brain regions that encode this information, is dedicated to the first memory, and these cells have certain biochemical changes that get them essentially ready for the next memory. One such change is an increase in excitability, so that when the second memory comes along hours later, the cells that were engaged on the first memory are more excitable and therefore more likely to also encode the second memory. With time, biochemical processes come along that essentially shut down this initial group of cells. So when another memory comes along, then it goes to a different group of cells. Okay, this is a really, really simple memory. A very, very simple model. And what I will do next is to share with you just a little bit of published data. I'll go very quickly through that, but mostly unpublished data. Focus on the role of mechanisms that shut down this linking process and separate memories across time. Now, for the first, all of this started with work that Chino Jocelyn and Yu Zhou did in the lab. And they showed that CREB-dependent increases in neuronal excitability. CREB is a transcription factor that's activated by memory. And downstream of CREB, there are increases in excitability. And this was critical to allocate memories to a subset of cells. And this was critical for memory linking. We later showed, together with Denise Kai. So Denise came into the lab and she set up a behavioral schema so that we could test memory linking in mice. So what Denise did was to train the animals in 3 different chambers, and then later she conditioned the animals in one of the chambers and then test them on all 3 of them. So, and what Denise found is that when she exposed the animals to the chamber that they saw 5 hours before the chamber in which they eventually were conditioned, what we surmised happened is that the animals recognized that chamber because they have a contextual memory of it, And then because that memory that they created 5 hours before the memory of the cage that they were eventually conditioned in, those two memories were linked. They thought of the cage in which they got shocked, and they froze, as the red bar on the figure shows. The amazing thing is that they froze as much as they did when they were shown the cage in which they were conditioned in. However, when they were shown the cage that they saw 7 days before, which we wouldn't expect, to be linked to the cage, to the blue cage in which they were actually, conditioned in, they showed much less freezing. And of course, there were many variants of this behavior, but this is essentially what the knees used to test, behavioral memory linking. Daniel Roney developed a set of head-mounted fluorescent microscopes that we have used in the lab. This is technology very similar to the technology that was developed at Stanford. And essentially what Denise and Daniel showed is that the overlap between the hippocampal CA1 representations of two memories that are linked, those ensembles overlap more than ensembles of memories that are unlinked, as you can see on by these two pictures on the bottom of the screen, the 5 hour and 7 days. So this was essentially what we had predicted in that earlier paper in 2009. So more overlap at 5 hours than at 7 days. And this was done with miniscopes. Denise essentially showed the same thing when she used immediate early genes in TET tag studies, where we can tag with immediate early genes the cells that are encoded by the cells, the ensembles that are encoding memory for A and B, either 5 hours or 7 days apart. And once again, you can see that there is more overlap between those ensembles, that the memories were created 5 hours apart than the ensembles that were created 7 days apart. Essentially 7 days apart, that overlap is what you get by random. And you get an increase, if you look at the ensemble overlap and memory linking, there is a progressive decrease in the amount of overlap that you see, as there is a progressive decrease in the memory linking that you can observe over time when Denise looked at the number of times in the same experiment that I just described a minute ago. Interesting enough, Kaoru Inoguchi's lab later showed that if he were to inactivate, this was a cool experiment, beautiful, beautiful experiment, that if he were to inactivate the overlap cells, it didn't affect the two individual memories, but it got rid of memory linking, which again was what we predicted by that model that we had proposed years before. So very consistent with what Denise found in her paper and with the model that we wrote. So what I would like to tell you here for the rest of the time, it's really unpublished studies about how is it that this comes to an end, right? So if all memories are linked and no memories are linked, this is useless. So memory linking is useful if indeed there is a temporal window, and I should say memory linking that's time dependent is useful if there is a temporal window in which you link memories and then after that you don't anymore. So, for example, in my case, I can link those memories that I created in Norway and separate them from the memory that I created later on, 7 days later. And what I will show you is that there is a molecular system that's engaged in those cells that were just activated by the first set of memories, so that later memories that come days later no longer recruit those same cells. So that when you recall memory A, you get memory B that was created hours apart, but not memory C that was created days apart. So you can link memories and you can separate memories. And this is a system that's based on CCL5 and CCR5, and I'll tell you about it next. CCR5 is a chemokine receptor. It's a G protein linked receptor. And the reason why we were very excited about it is that CCR5, we showed in a number of papers as a negative role in MAP kinase CRAB. Why is this important? Because this has to do with memory and learning and structural plasticity and synaptic plasticity, and of course memory, that we could manipulate all of these mechanisms and memory itself by manipulating CCR5. So we thought that this may be a good candidate for a molecular system that shuts down memory linking, because it's shutting down memory on those cells that were just engaged in linking. So you separate memories across time. Another thing that we showed in the lab recently, Giselle Fernandez in the lab showed, is that CCR5 also manipulates excitability. Why is this important? Because excitability, we showed before, is one of the key mechanisms that you get that overlap. And when you activate CCR5 with its ligand CCL5, you decrease that excitability, so you put those cells offline. So CCR5 has a negative role in a bunch of things that are critical for memory, as I mentioned already. So, but are the levels of CCR5 dynamic following learning? And the answer is yes. Both CCR5 and its ligand CCL5 are dynamically modulated over time. In this case, we are looking at RNA expression. With a delay of hours, you see an increase in expression of this system that then shuts down memory on those cells that were just engaged, so that you separate them from other memories that come later on. We wanted evidence, direct evidence, that this is actually taking place during learning, that you are activating this receptor system. And to do that, we took advantage of this beautiful system that's optogenetically activated and that was developed by the Quan lab. The idea is that Yang Shan in lab designed a sensor, a molecular sensor for CCR5 activity that allows us to see it in real time as animals are learning, actually, to be more specific, following a learning experience. And what we predicted is that we should see an increase in activation of this sensor, this molecular sensor, which reflects release of CCL5 and activation of the CCR5 receptor, which we know blocks memory. And so there are multiple parts, to this beautiful system that the Quan lab designed. Ultimately, it's gated by optogenetic activation, and when the right circumstances line up, specifically when the ligand is sensed in the environment, then you get activation of GFP in the nucleus by a transcription factor that's released when the receptor is activated. It's really a beautiful system. And what Yang Shen showed is that indeed he could see a gradual activation of this receptor system following learning at very specific times that he measured by optogenetic activation of the sensor, which is really nice. So you choose when you get to make measurements by optogenetic activation of the sensor, as I showed in that cartoon. The amazing thing is that the cells that were just engaged in memory, and Yang Chen could tell which cells those were because they had immediate early gene expression, those cells showed more activation of the CCL5, CCR5 system, which is consistent with the model that we have been sharing with you. All right, one of the key predictions of the model is that if you knock down CCR5, you should change which cells are engaged in memory. Because remember, when CCR5 comes up, those cells are avoided. What about when you knock down? When you knock down, those cells now are preferentially engaged, as this result shows. So if you knock down CCR5, those cells are preferentially engaged in memory. What about the opposite experiment? What about if you activate this receptor system? Can you block memory linking? Because you'll expect eventually this molecular system comes on, and days later, then those cells are not engaged anymore, you block memory linking. What about if you activate that system at a time when you normally would see memory linking? And what this result here shows beautifully, and this is work by Andre Souza and Yang Cheng, is that you can shut down memory system. So, that you can shut down memory linking by using this system. So you can manipulate where the memories go with this system, and you can manipulate whether they are linked or not. And you can do this with the knockout too. You can do this with the knockout too. You would expect that if you get rid of the break of memory linking, you have extended memory linking. And that's exactly what Mio Zhou showed in the lab. When he knocked out the break of memory linking, he now could see memory linking even at 7 days. When we looked with miniscopes and looked at the overlap, essentially what Mio Zhou showed, by knocking out the thing that stops memory linking, the thing that should stop the overlap with time, let's say at 7 days, that's shown in this figure, now it didn't see it, and you see it very nicely here, that at 7 days, the knockouts, the CCR5 knockouts, have as much memory linking as the wild types do at 5 hours. I should
actually, no, it's probably time to wrap up in a few minutes.
Yeah, sure. And so in essence, this is, the progressive decrease of memory linking, and we can actually optogenetically activate, CCR5 and turn down, memory linking with a hybrid system between the rhodopsin and the CCR5 receptor. And here we can show that by activating this receptor at specific times, we can shut down, memory linking. And this is optogenetic activation of this receptor. I don't have time to show you this in this talk, but the interesting thing is that with age, there are increases in this memory linking break. And interesting enough, you lose memory linking, but when we get rid of the break in aged mice, we restore, memory linking. So CREB has a critical role in opening the window for memory linking. CCL5 has a critical role in closing that window. And with aging, the window is closed, but we can reopen it by manipulating this molecular system. And I mentioned these people as I went along. And the last thing I want to say is that there are wonderful opportunities with these new cell type and circuit-specific molecular tools to open windows into circuit and behavioral function that you otherwise would not have. Thank you.
Thank you very much. That was a wonderful talk. We had a few questions from the audience. The first one, Alcino, is what if, this is from Francine Dollins, what if environments are not discrete? Does the animal have to cognitively parse that sub-environment within the larger space?
That's a beautiful question, and it's one that allows me to clarify an important point. Animals have many ways of structuring multiple memories. One is by simply using time. In my example, those two scenes were radically different. One was a scene of ocean, the other one was a little village. And nevertheless, I can put them together because they happen within the same window of time. But you imagine that memories can have common elements. And when they have common elements, they can be linked into the same memory structure. So you have multiple, the brain has multiple strategies to create memory structures and to relate memories that we acquire at different times. I just spoke about one, using time as the entity that links memories. So, going back, to your question, of course, if the cages are similar, then the animals are going to link the memories that were created in those cages because they have the cage as the linking element. I hope I answered your question, actually.
It was Francine's question, but I think that was good. Thank you.
Yeah, I meant Francine. I was addressing Francine.
Oh, okay. This is very unusual, having a face-to-face conversation while 500 people are watching you.
Yes.
There was a question by Timothy Behrens. Are you changing the activity of the cells when you manipulate CCR5?
That's a wonderful question. And the answer is yes. We actually tested that, directly. And what Giselle Fernandez showed in our lab is that by manipulating CCR5, we can manipulate excitability. But again, remember, we are directing our changes or we are directing our observations to a specific group of cells. So it's not all over the animal. It's in a region of the brain that we showed is critical for processing the information that we are giving the animal, in this case, contextual information, and in a region of the brain that we showed is essential for linking these memories, because when we either artificially overlap memories there, we can link them, or when we artificially disrupt the overlap there, we can unlink them. So the answer is yes and no at the same time. I hope that was helpful.
There was one question from Andre Fenton. The question was, how do you think about the role of the role of specific synapses for linking / separating memories within these beautiful cellular level data to support the idea of a cellular allocation to a memory? Do all the cells synapses?
Great question. That's a wonderful question. And it's really wonderful because it's the work of Mega Siegel in our lab. So we actually develop a set of technologies that allow us to query which synapses are engaged in which memory, where those spines are with 2-photon, and then see how dendritic segments and spines are themselves allocated and what are the rules by which memories are linked or not. And that's an entire different talk, but this is just a long way of saying that we are very much aware that memories are not stored in cells, obviously. Cells are just our tag to follow those memories ensembles in the CA1 with head-mounted fluorescent miniscopes or 2-photon or whatever. But the idea is that on a separate talk that I hope to give some other place, some other time, I can tell you about what we have learned about the role of spines and dendritic segments in the process of allocating and linking memories across time. Wonderful question. Will take too long to answer, unfortunately. Sorry. Thank you, Andre. I really appreciate it.
That was good.
Very, that's a really important question. Yes, absolutely.
All right. Well, thank you very much, Alcino. This is wonderful.
Thank you, guys.
I think we're going to wrap up there. Yeah, thank you for coming. Thank you to all the speakers. This was an absolutely wonderful session. I truly enjoyed it very much. I found it very inspiring and fun, frankly. Before we wrap up for today, I wanted to give you a preview of what is coming tomorrow. If I can pull it up on my screen here. Hopefully everybody sees this. So we're going to wrap up for today. What you are seeing on the screen here is what you would find if you go to inavsymposium.com. This is the what we have planned for tomorrow's presentation. Sorry, this is Tuesday. This is tomorrow's presentation. That was today's that you were looking at on the screen there. Tomorrow we're going to start at the same time, 11 o'clock Eastern time, 5 p.m. European time, 8 a.m. West Coast of America time. And we'll start with the data blitz session for the first hour and a half, followed by a little break, which will be my lunch break, for example. But different people have different things. And then we'll have session 4 with Jennifer Bizon, Jennifer Campos, Jan Wiener, and Paul Smith following after that. And the last thing that I want to tell you about today is that if any of you missed any talks, or if anybody in your lab wants to miss any talks or anything like that, all of the sessions are recorded here at iNAV, thanks to the Crowdcast system. And you should be able to access the older sessions. My understanding is by logging into Crowdcast, if you click in the upper left-hand corner and click on schedule, and if you click on one of the sessions for one of the previous sessions that have already happened, you should be able to download or stream the videos from one of the previous sessions if you missed them or if anybody in your lab is interested in them. So that's a very fun feature. I believe that's it for today. So thank you very much. We'll have the Data Blitz session tomorrow to start. If you haven't sent in your Data Blitz presentations, please do so. And if not, everybody have a good rest of your day, and we'll see you tomorrow. And that's the end of iNAV Day 1. So thank you very much.